Hello students, welcome back to the biology class. So we are doing chapter number 13, photosynthesis in the higher plants. In the last class we did the topic of light harvesting complex, and we did the differences between photosystem first as well as photosystem second, and we started somehow the topic of the light reaction, and we understood what is photophosphorylation and its two types. We did in the last class photosystems. We understood that it is the smallest number of pigment molecules which need to function together, and Begins called it as quantosome. There are other names like light harvesting complex, photosystems, pigment systems, and we learned that each such system is made up of one reaction center made up of chlorophyll A molecule and harvesting molecules which are supposed to pick up the sunlight and transfer to the reaction center. Harvesting molecules are of two types. All chlorophyll A molecules will form core molecules, and chlorophyll A, xanthophylls and carotenes will form the antennae molecules. We did the differences between photosystem first and second. The main difference is the reaction center, maximum absorption 700 nanometer and 680 nanometer, and it is situated on the outer surface, inner surface, in the stromal lamellae and non-oppressed granule lamellae. Only in the oppressed granule lamellae. This is involved in both cyclic and non-cyclic, but this is involved only in the cyclic type of photophosphorylation. In this photosystem is involved with the reduction of NADP into NADPH2, and this photosystem is involved with the splitting of the water. We understood in the last class that the process of photosynthesis is divisible into two steps: oxidation of water. And we all know, because redox reaction occurs simultaneously, there is oxidation as well as reduction. So once the oxidation of water takes place, that is called as the light reaction, it liberates oxygen gas as byproduct, and H, because loss of hydrogen is called oxidation. So the H or you can say protons which are released are picked up by the NADP, and NADP reduced into NADPH2. This NADPH2 or NADPH would go into dark reaction, during which the NADPH2 will liberate the H, and this H will go and interact with the carbon dioxide, or you can call it as reduction of the carbon dioxide to form glucose molecule. This occurs in the dark reaction. We started the details of the light reaction in the last class. That light reaction is a light dependent, that means directly dependent on the supply of the light duration, light intensity, light quality. Quality means the vibgyor, which wavelengths are falling. All these things play a major role in the light reaction, and the light absorbed by the harvesting molecules, it means core molecules and antennae molecules, will be transferred to the reaction center, and reaction center will become excited, and because there is magnesium atom there, it will liberate the electron pair. These electron pairs run through the electron transport system, and finally ATP and NADPH2 are produced. This formation of these compounds is called as photophosphorylation. During this process, breakdown of water takes place in the presence of light and manganese as well as chloride as well as calcium ion. This was explained by Neil and demonstrated by Robert Hill, and in this process water splits into H plus and oxygen as well as electron. H plus accumulates in the lumen. Its benefit we'll be learning in the next class, and oxygen will go out as the byproduct of photosynthesis, and electrons are transferred to the photosystem second. So we started in the last class regarding the photophosphorylation, that it is the process of synthesis of ATP with the help of the light energy, and it was discovered and explained in detail by Arnon and his colleagues in 1954. The formation of ATP in the presence of the sunlight is called as photophosphorylation, and this occurs in two methods. So there are two types of it. One type is the non-cyclic and the other type is the cyclic type. The non-cyclic type involves the non-cyclic transfer of the electron. So electron never comes back to its source of origin. So because electron never comes back, it is called as non-cyclic transfer, and ATP is formed in this process. Along with ATP, even NADPH2 is formed in this process, and both photosystem first as well as second are involved in this, and there is splitting of water. So the splitting of water liberates electrons which are picked up by photosystem second. Photosystem second will lose electron which will go to photosystem first, and photosystem first will lose its electron to NADP. So that means electron never comes back, it is gone to the NADP finally, which turns into NADPH2. That's why it is called as non-cyclic one. And the cyclic photophosphorylation is the formation of ATP during the cyclic flow of electrons. So when we say cyclic, that means electron will come back to its source from where it has started, and then it will involve only one system, and that is the photosystem first. And there is no reduction of NADP into NADPH2 in this process. Only ATPs are formed. Here both are formed. In this case only ATP is formed, and there is no splitting of water. So that means there is no oxygen liberation associated with the cyclic photophosphorylation. So let's quickly go through this. NCERT does not explain these details, so it's your wish whether you want to go through detail or just stick to the NCERT. So when sunlight falls on the plant, photosystem second also will receive sunlight, even the first will receive sunlight. So the photosystem second will be excited and will lose the electron to the pheophytin, that is the primary acceptor of the electron, which will transfer the pair of electron to plastoquinone, and plastoquinone will transfer the electrons to cytochrome B6 and F complex, and finally to plastocyanin, which will transfer the pair of electron to photosystem first. And photosystem first also liberates electron because of excitement, which transfers the electron to the FeS, that is ferredoxin, basically iron sulfur rich compound, ferredoxin, and this will transfer the electron to the enzyme which is called as NADP reductase enzyme. So this electron from here will go to NADP, and finally NADP will turn into NADPH2 because of this enzyme called as NADP reductase enzyme. In this process, sometime FADH2 also is changed into FAD. So both concepts are possible. Electron never comes back. This is the main point here. In this concept, photosystem first is only involved. It loses the electron to the primary acceptor, which goes to the cytochrome B6 F complex and plastocyanin and comes back.